get started with introductions, maybe?
I think that's a great idea.
Okay, disclaimer first, though?
Sure. Do you want me to read it?
Yes, please.
Okay. Hi, everyone. The information provided in this town hall is presented to provide general education and information. The information provided is not medical advice and should not be used as a substitute for the advice of a physician or other qualified health professional. The information provided by presenters or commentators is believed to be accurate as of the time presented. Any opinions expressed by presenters or commentators are those of the persons presenting or speaking and are not necessarily those of the American Society for Virology, the American Society of Microbiology, the University of Virginia, the University of Michigan, or other sponsors of this education initiative.
Okay, Brianne, do you want to start? Introduce yourself?
Sure. Hi, everyone. My name is Dr. Brianne Barker. I am an immunologist at Drew University, and I have been working on immune responses to vaccines for more years than I'd like to admit, and I'm excited to answer all of your questions.
I'm Angela Rasmussen. I am a virologist. I'm currently an affiliate at the Georgetown Center for Global Health Science and Security. Shortly, I'm moving to Canada to be starting a lab at Vaccine Infectious Disease Organization, International Vaccine Center, Vito Intervac at the University of Saskatchewan. It's a really long title for my institution. I study the host response to viral infection for emerging viruses, including SARS coronavirus 2.
Okay. I'm Dr. Jason McClellan. I'm a professor at the University of Texas at Austin. I'm a structural biologist. My lab has helped determine the structures of the SARS coronavirus spike proteins. We actually engineered the protein to help keep it more stable. That engineered form is in most of the vaccines that are licensed in the US. And look forward to chatting with you all.
All right. Do we have anyone with some specific questions? If you want to raise your hand or send them to everyone in the chat, that would be great. We've had some other questions pre-emailed in, and so we can go with those as well if no one has any other questions that they're burning to have answered right now.
We have two in the chat.
Okay.
I'll read the first one, and you can weigh in. For 20-something women who have already had COVID-19, should they get the vaccine? Is there data yet to show that the vaccine is safe for those who may become pregnant in the future?
All right.
I can start.
Go for it.
So the vaccine has been administered to many women many people who are pregnant. And so far, it doesn't look like there are any negative side effects associated with being vaccinated during pregnancy. In addition to that, there's data not in pregnant people, but in a general cohort of people who've had COVID before. And it looks like vaccination with even one shot of the vaccines that are available can really boost those neutralizing antibody titers that are thought to be really important for providing protection. So all people who've had COVID-19 before should consider getting a vaccine, given that it's likely to improve your immune responses, improve immune durability, or how long the vaccine can remain protective, as well as pregnant people should definitely consider getting vaccinated.
I think that's a great point. The natural infection leads to a pretty wide range of different immune responses, from almost a negligible immune response if it's a very asymptomatic infection to a really strong immune response for people who are hospitalized. Whereas the vaccines produce a pretty uniformly robust response. And so just because you've been infected previously, we don't really know what your titers are, how strong your immune response was. So we really think you need to get vaccinated and get your neutralizing antibody titers and immune response as high as possible.
Yep. And so if you've read things that have made you have this concern in the past, there was some information about some kind of effects on fertility or things of that nature because of similarities between the protein encoded by the vaccine and a protein involved in the placenta. If you actually look at that level of similarity, it is really not particularly similar. It is sort of at the level of similarity that you might expect if I decided to write something and Shakespeare decided to write something, and we noted that he and I use a couple of the same words. And so it's not really a level of similarity that indicates that there is going to be any kind of immune response that's going to cross-react.
The second part of the question was, are there any data on how many spike proteins are produced as a result of the vaccination? Does the amount of spikes expressed vary a lot person to person? There's no data on that in people to be able to look at the number of cells that get transfected with the mRNA, the number of spikes that each of those cells produces. But again, we do know that it's a very uniform response to the vaccine, suggesting that most people are producing a similar number of spikes on average.
That's great. Have you had other questions come to you in the chat? I've had a couple, so.
Yeah, I've had direct, go ahead. Yeah, I have some too. Brianne, go ahead.
I do too.
Okay, great. So tonight, what are the differences used
Brianne, you're
production of vaccines in the efficacy and protection based in the, oh, okay, I'm sorry about that. Am I okay now?
Yes.
Okay, great. So this question is, what are the differences between antigens used in the production of vaccines in the efficacy and protection based in the viral cycle?
I think I can take this one, if I got that. So the coronaviruses have many different genes. Many of them, we know what they do. Some of them are actually still investigating. But the vaccines, the Pfizer and BioNTech, Moderna, Johnson & Johnson, AstraZeneca, all those are just delivering the spike protein. So only one protein from all the different SARS-CoV-2 proteins. So we really focus the immune response onto that protein and generate really robust antibodies that can prevent the function of the spike. So the spike is critical. It's the little white dots on the surface of the virions behind me. It has two functions. It attaches the virus to our cells by binding to specific receptors on our cells. And then after attachment, the spike undergoes this really huge conformational change, like this morphing between the initial conformation and this final conformation. And antibodies can also block that. So we really want to try and coat all the spikes with antibodies, prevent the virus from infecting our cells. And that's why we just target the spike protein in these vaccines.
Yeah, and I can add to that just a little bit. When you are infected, normally, as Dr. McClellan just said, there are a bunch of different proteins that the virus will make. Normally, you will mount an immune response to several other of those proteins, what are called the structural proteins, the N protein, the M protein, and the E protein, in addition to spike. You know, it's not really known if that can enhance or improve the immune response, but certainly the spike protein is critical for the virus to be able to infect cells, and neutralization via this is thought to be very protective. In addition to that, there are other proteins, as we just mentioned, including some that we don't know the function of, but the function of some of them is likely to evade the host innate immune response, which can actually dampen those antibody responses and the T cell responses that make vaccines work. So by giving just the spike protein, you're taking away the activity of all those other potentially immunosuppressive proteins that might lead to immunity that is not as robust as you would get from just getting the spike protein.
That's great. And one thing I will add is that one of the great contributions that Dr. McClellan has made in this field is understanding some parts of the structure of the spike protein and some changes we can make in the spike protein in order to make a better vaccine antigen. And so the vaccine antigen has been in a way that is it and allow for a better response. You are in fact speaking who figured that out.
Brianne, your audio is kind of cutting in and out.
Okay.
Yeah, I don't know if you just try like turn off video and just go audio only, or. Angela, thoughts?
I don't know. Sometimes turning the video off can help. I actually have two speeds of wireless, and sometimes switching from one to the other will help, especially if my spouse is on his own Zoom call. Um, but yeah, maybe going dark, Brienne, will help.
Well,
Um,
let's hope so.
Otherwise, there's always the turn it off and turn it back on
True,
approach,
true.
which is one of my go-tos as well.
Yes. Let's see if this makes any difference.
So far, it's better.
Great. So I have lots more questions have come in. I'll do one more and then we can sort of rotate around. So the next question I have is, are females at more risk for side effects from the vaccine? The news would have you think that, but I also realize they highlight only a very small percentage.
So I can sort of address that. To my knowledge, there's no data suggesting that there are sex differences in terms of the side effects that people experience when they get the vaccine. But it is well known that women and men will have different responses to both infection and vaccines. They have fundamentally different immune responses, largely. I mean, there's a combination of factors, but it's thought to potentially be mitigated or influenced by sex hormones like estrogen and testosterone. So it is entirely possible that men or women might experience differential side effects, but I don't know. Correct me if I'm wrong, Brianne and Jason, but I'm not aware of any data that has really looked into that deeply.
I would say
Uh-oh, Brianne, we lost your audio again.
Oh no. Okay. Um, so I will say that I agree. There is no specific data on that. Um, but there is sort of some known differences in the immune responses between men and women.
Yeah, and there is a very clear sex bias in that men are much more likely to have severe disease and die from COVID-19 than women. But as far as I know, there's not really much characterized so far about sex differences in vaccination.
Angela, do you want to read a couple that you've had?
Sure. Here's the first one that came in for me. I understand mRNA technology has been around for a while and has been used in clinical trials, like for the flu virus and cancer. But why is COVID the first vaccine to receive some type of approval? What happened to the other mRNA clinical trials?
My let me know if I tried to make a change, so let me know if that improves my audio. So my simple answer to that is money. There were other trials, particularly for vaccines against things like rabies, CMV, and a couple of others. But each stage of the process takes some funding to kind of start that and requires a fair amount of interest and impetus in making that vaccine. And because of all of the effects that we have seen with this pandemic, we really prioritize putting the funding towards this and making sure that we did those steps quickly. Some of those other vaccines have been useful, but just were not pushed forward in the same way.
Some of the vaccine.
Oh, go ahead. Go ahead.
So some of the vaccine, I think Moderna's probably had close to 20 different clinical trials on different mRNAs. For some, they're trying to make vaccines for which there already are pre-existing vaccines. So the bar is higher. You have to do better than what's currently available. Some actually look really quite promising. It takes 5 to 10 years to go through all the different steps of the process. So for coronavirus, we had something where there was no pre-existing vaccine. We had an urgent need for it. The technology worked really well. It was very well suited for pathogen response. And that's why it was what the two first to the finish line were the mRNA vaccines.
Yeah, and I was just going to add a brief. One of the vaccines that Moderna had developed was a MERS coronavirus vaccine, and MERS coronavirus is very serious, but it also occurs sporadically. There can be outbreaks of it, but they're not very large, so it's hard to enroll the large numbers of people that you need for later phase clinical trials when you have a pathogen that doesn't happen very often. In addition to that, just adding to what Brianne was saying, this is a common story for vaccines for emerging pathogens, is that they tend to get caught up in the development cycle before really moving forward to the finish line in clinical trials. And a quick story I'll tell is the Ebola virus vaccine that's manufactured by Merck and got approval in December of 2019. That vaccine was actually developed 10 years before the West African Ebola epidemic. It's a different type of vaccine, viral vector gene, but basically it had been tested extensively in animals, but there weren't huge Ebola outbreaks happening, and there really wasn't a large incentive for any manufacturer to bring that forward through the clinical trial process. So it wasn't until there was an emergency with the 2014 West African Ebola epidemic that moved past the preclinical and early clinical development stage. So this is a common theme, and it's something that I think going forward we should probably be rethinking in terms of the cost and the efforts required to get vaccines approved.
Angela, do you want another one?
Sure. Okay, so I have somebody who sent me several questions. And actually, I don't know the answer to this one, so I'll be curious to hear what you both have to say about it. I saw a TV news report from Florida that says the arm shouldn't be squeezed when injecting the vaccine. Can you tell us?
Angela, you broke up there. I think you were saying something about the arm being squeezed.
Okay. Yeah, the arm shouldn't be squeezed when you're getting the vaccine injected. And is this something that we should be concerned about? I have not heard that. I imagine that that's probably just because you shouldn't be squeezing somebody's arm when you're putting a needle into it. But I'm curious to know if this is anything you've heard about.
I have personally not heard that. I can't imagine that it was actually controlled in some sort of study to determine whether squeezing or not squeezing had any effect on efficacy. So I haven't seen that study, but I wouldn't be that concerned about it.
I have not seen that study either. I wouldn't be that concerned about it. If I were trying to come up with something in terms of a scientific mechanism, I will say that we know that these mRNA vaccines are best delivered intramuscularly. And we don't want to deliver them into the skin intradermally or into other tissue layers. We want them to go intramuscularly. So I suppose if you're squeezing somehow influenced which layer you were injecting into, perhaps that would be a problem. But I don't think that that is an issue that we're really going to be dealing with in the real world.
And here's the second part of this question, which is actually not related at all to the first, but I think it's a good question. And then I'll pass it back to you, Jason. After someone receives a vaccine, can they still take an antibody test to find out whether they ever had asymptomatic COVID in the past, or does the immune response to the vaccine make that no longer possible? And I think this is an incredibly useful approach that doesn't get discussed as much, and it's relevant to something we were talking about before, and that is the difference between immunity to the vaccine versus natural infection. As we were discussing before, when you're naturally infected with the virus, you mount antibody responses to all of these other viral proteins in addition to spike. So you can actually go back and do an antibody test looking for anti-N antibodies, which you will not have if you've only ever been vaccinated. So you can determine if you had asymptomatic or pouchy symptomatic COVID in the past by doing a serology test like that.
And then just note, Angela, though, that it would depend very much on the type of test specifically that was being administered. Some contain both spike and N. So although academically you could do it, it would depend very much on the kit or whatever it was being used for your particular case.
That's absolutely right. If you wanted to actually do this yourself when you go to the doctor, you make sure that they're ordering a test that's specifically anti.
Yeah, so no spike in the test.
Exactly.
All right, cool. So I got a couple of questions. One is concerning the difference between J&J and AstraZeneca and the science behind those. So those are both pretty similar in that they use an adenovirus, which is a harmless type of virus. The Johnson & Johnson uses a human adenovirus 26, Ad26. They just insert the spike protein into that virus's genome. You inject the virus into the muscle. The infected cells produce different viral proteins, including that spike gene that was inserted. So Johnson & Johnson is a single dose, Ad26. The AstraZeneca is a chimpanzee adenovirus. So it's a chimp ad, and it also contains the spike gene inserted. There's slight differences between the spike sequence that are used in the two vaccines. The Johnson & Johnson contains the two stabilizing mutations that my lab developed. AstraZeneca doesn't. I don't think that's too big of a deal. I think there's some slightly smaller differences. The AstraZeneca does have prime and boost, and yeah, that's probably it. For AstraZeneca, there's been some concern over the vaccinations being halted due to potential links to blood clotting, whereas we haven't seen that with the other vaccines. I think that's something that's still very much under investigation. A lot of different manufacturers are making the AstraZeneca vaccine, so potentially it could be a batch difference since there's no blood clotting issues observed during the large phase 3 clinical trials. I think it's still relatively small number, like 1 in 100,000 people. So whether there's a true cause from the AstraZeneca vaccine or not, but so far I wouldn't be concerned about getting either the AstraZeneca or J&J. And Brianne and Angela, if you have any thoughts to add on that.
I would agree completely with what Jason has said here. I think the one thing I would just point out for those of you who are in the United States, the Johnson & Johnson vaccine is approved for use in the United States, and so it is one that you may get. AstraZeneca right now is not approved in the United States, and so it may not be one that is available to you at the moment.
Yeah, and I would only add to that too that while the side effects, the thromboembolic blood clotting events that Jason was referring to have been reported for AstraZeneca, they're still incredibly rare, and you actually have a higher risk of having a similar severe blood clotting issue by taking oral contraceptives. So keep that in mind. It's not as though one of these vaccines is for sure going to cause these serious problems. Like Jason was saying, we don't actually know that they're even related to the vaccine, and they might be a batch specific issue. Adenovirus vectored vaccines have been used for a very long time in a number of other clinical trials, and they're generally thought to be very, very safe.
One question, what was done to augment the structure of the spike proteins in order to make the spike proteins easier to be used for a vaccine? So the spike has to change conformations. One part of it particularly has to go from like a bent spring to an elongated spring. We changed a couple of residues in the hinge between the part that has to spring to kind of rigidify the protein, prevent that from happening. And well, we did that back in 2016 and showed that it worked really well for the MERS coronavirus spike, the first SARS coronavirus spike, and seems in general to work well for beta coronavirus spikes. The effect that those stabilizations have on efficacy, we'll really never know, but we think it helps. We've made a second generation spike that contains a few additional stabilizing mutations. That also looks pretty good. A phase 1 clinical trial was started in Vietnam 2 weeks ago and in Thailand soon. So those are some of the small changes just to try and improve the expression level of the spike protein so you get more spike per infected cell, as well as the stability of the spike to keep it in that conformation longer.
It's great to have someone who knows so much about those exact changes here to answer that question.
I know.
So this question is sort of two parts that are related, and so I'll put the two parts together. The first part is there a significant difference between the Pfizer and Moderna vaccines? And the second part is, could you describe briefly how the body responds to mRNA vaccines after they are taken, and what happens in the time it takes for a full person to become fully immune after vaccination?
I think I can handle the differences between Pfizer and Moderna.
Excellent.
Yeah, okay. So they're both mRNA vaccines. Pfizer has released the exact sequence, the recipe of the mRNA in their vaccine. So we know the different, what are called codons, exactly which nucleotides are in there and how they're modified, so that they have a non-natural nucleotide in there. And so we know that. We know the sequence of the spike protein. The sequence of the spike from Pfizer and the sequence of the spike from Moderna are very similar at the protein level, but we don't know Moderna's the nucleotide level, or at least not released by Moderna. A group did sequence it recently, so we have that information. We don't know about the type of modifications, exactly which types of nucleotides they're using in the mRNA and how they're modified, and if there's different modifications from Pfizer's. And then another part that we don't know specifically is the lipid nanoparticle that's used to encapsulate the RNA and help the RNA get across our cell membranes and into the muscle cells. So that we don't know the specifics for the Pfizer and the Moderna. So at a protein level, the proteins those vaccines are going to produce essentially identical. The mRNA is very similar, about 91% identical. Lipid nanoparticle, I don't think we know that much about.
So some of the information on the lipid nanoparticles has been released. And so if you look at the product information from both Pfizer and Moderna, you can see a list of ingredients. So if you are worried about, say, allergies on that front, you can get a list of which particular ingredients are in there.
So I think you don't know anything about like the ratios or
exactly.
Yeah.
Which I would love to know, but in terms of understanding it, if I have an allergy, that information is out there. So those lipids are really important because they will help that mRNA get into cells. The mRNA can get into a couple of different types of cells. There's some debate in the literature about exactly which kind. I'll say everybody's right. Either into some of the muscle cells at the site of infection or some immune cells at that local site. In any case, whichever cell is getting that mRNA, the mRNA has been helped into the cell by the lipid, and that mRNA will go straight to be read by part of the cell called the ribosome to be made into proteins. mRNA is usually sort of a message that's carried around in our cell. It's not something that's made to kind of be permanently sticking around. It's sort of like the Post-it note that you carry some sort of small message on that's not your main text. And so that message, mRNA, will get translated into protein. And so we'll make lots of copies of that spike protein in whichever cells have received that mRNA. So this is going to be some small number of cells, either immune cells or muscle cells, at the site of the vaccine. It's not going to be all of the cells of the body that suddenly get this new mRNA. That small number of cells will make the spike protein. That spike protein can be put on the surface of the cells. It also can be processed by some immune proteins in the cell and put on the surface after processing. And those self cells that are making spike can now turn on antibody and T cell responses. And so we'll have your body start to make immune responses with both antibodies and T cells. The antibodies will be able to protect you against the virus, but you'll also make some cells that are called memory B cells that live for a long time to make antibodies long after that initial infection or that initial exposure, as well as some memory T cells. And so that's kind of the general part of this process. Those cells that are making spike are probably going to die relatively shortly after this process. They may be killed by immune responses. So you may, lose a few cells, not a whole lot. You're not going to have them permanently making spike throughout your body and wreaking any sort of havoc. And so that's kind of the general idea of how the immune response happens with this mRNA vaccine.
Great. I have some more, but Brianne, do you have any questions?
I have a lot.
Okay. All right.
I do too.
I'll do one more and then we can go, then we can switch.
Yeah. And I think we want to touch on the variants at some point. I don't know if we have some questions about that.
Yeah.
I do not have any questions about the variants. So I guess the one question that is related to what we've been talking about before is this question about the J&J vaccine, Johnson & Johnson vaccine, whether it is protecting against asymptomatic infection like the mRNA vaccines, and whether this person could get boosted with, can get J&J and then get boosted with something else, whether they have to worry about getting other adenovirus vaccines in the future. They have a few questions.
I mean, I think that certainly vector immunity, which is really what that is a possibility. And we already know that from other adenovirus vectored vaccine trials that certain adenovirus vectors that are more common in the human population, adenoviruses typically cause common colds, can lead to more people having antibodies against those adenoviruses, and that will lead to the vaccine itself being cleared before it can go in and infect cells and start making the spike protein. Ad26 is an alternative because it's not as common in the human population, so most people don't have pre-existing immunity to Ad26. That's the same reason why the chimpanzee adenovirus used by Oxford and AstraZeneca was also chosen. That's also probably why the Sputnik V vaccine developed in Russia uses what's called a heterologous vaccine regimen, where they're using two different types of adenovirus to vector the prime shot or the first shot followed by the booster dose. So probably over time, yes, you probably can't repeatedly vaccinate with the same adenovirus vector. But I don't know that there's actually a ton of data on that. I think that probably in some people, depending on when those vaccines are given, if they're given years apart, it might not be such a problem. If you're trying to boost over and over and over again in a short period of time, it might be. So that's something that I don't think we really have a square answer on. But this is why it's great that we developed so many different vaccines, because certainly heterologous boosting with other vaccine technologies, for example, you get the Johnson & Johnson shot, and then you need a booster, it turns out, and you can get an mRNA vaccine or a protein subunit vaccine, such as the one made by Novovax that's currently in phase 3 clinical trials. So I think that there's a lot of options, and that vector immunity isn't going to completely ruin COVID vaccines for us. But I'd be very curious to hear Brianne and Jason's perspective on that as well.
Yes, so I've seen some animal data on Ad26 responses, which is the adenovirus type that was used by Johnson & Johnson. And Ad26 seems to lead to relatively short-term anti-adeno responses. And so it does seem, at least in animal models, that multiple administrations are possible because you don't have an anti-vector response. And that does seem to be unique with Ad26, but I have not seen those data in clinical trials.
Actually is also worth mentioning that I left out is that these adenoviruses are replication incompetent. They've had parts of their genome deleted that make it so that they aren't going to make more adenoviruses when you get these vaccines. So they're not going to be making a ton of the proteins that your body would recognize to make anti-adenovirus immune responses. They're not necessarily going to be as much of an antigen as the spike protein would that they're encoding. Okay, so I have two questions that I'm going to combine, and one of them starts to get at variants, even though it's not directly about that. So the two questions I'm combining is, what is the importance of the second dose? and that I'm combining with, do we have opinions on how feasible it will be that vaccine-induced antibodies will generate escape mutants to a scale that's significant for public health? And I'll let you guys start off with this, but I thought those two things are somewhat related.
Sure. So if you look at the data that was published from some of the early phases of Moderna and Pfizer's trials, there was a big difference in the antibody response after that second dose. There was good protection seen relatively early, but there was a pretty dramatic difference in antibody levels after the second dose. You could imagine that might influence things like how those memory cells were being made or longevity, though we don't have officially any data on that. I just like to, geek out about that part. And I'll say one other general thing, and I apologize for any of you who have heard me on a podcast before, because this is something I sometimes talk about pretty often there, which is that your immune response actually involves a lot of different types of cells and molecules, antibodies, but also a number of other things, including some cells called T cells. And there has recently been some data published indicating that T cells seem to not really have any changes in their responses based on when they're responding to the variants. So the T cells don't seem to really care about the variants. From the data I've seen, maybe the variants might evade antibodies a bit. And so maybe the incredibly high level is going to be very helpful, whereas if you had a slightly lower level, that wouldn't be great. But happily, we also have this T cell response that is going to protect us and that doesn't seem to be affected by the variants.
Yeah, and I would just add to that because, when the immune response is happening, it's not just an antibody to one part of the spike protein. Your immune response will make antibodies to the entire spike protein. And so if you think of the spike protein as sort of like a three-dimensional spike, it's not just like a triangle, it's like a cone, then you're going to have antibodies binding all over the surface of that. If there's one or two mutations that may reduce neutralization, but as Brianne was just saying, there's a huge repertoire of T cells as well that will also be responding to that. So it really is a very high bar for any variant to clear to get around those types of antibody responses. And given the totality of the immune response, B cells making antibodies, there's also protective non-neutralizing antibodies that we really don't know that much about right now for this virus. It's going to, I think, personally be very difficult for completely vaccine resistant variants to emerge. Now, that said, we of course should be concerned about variants, especially ones that will be able to continue to spread even if they're not causing severe disease, because that gives the virus more opportunities to make new variants and to continue to evolve around our defenses. But I don't think that if people are vaccinated, these vaccines are highly effective in real world situations. We just saw a study from the CDC this week that showed that vaccines significantly reduce infection, or at least the mRNA vaccines significantly reduce infection as well as reducing disease. That then we really should be in good shape, that there won't be opportunities for variants to emerge if enough people do get vaccinated.
And I'll take maybe a slightly different spin on the variants, that it does depend very much on which variants. The UK variant isn't really able to escape the vaccines as well as the variant from South Africa. That's probably one of the major concerns where we see the largest decrease in antibody neutralization and even in efficacy, because some of the vaccine trials, the trial sites were in South Africa, and so we've been able to compare differences to the vaccines, get the trial that occurred in the UK to the trial that occurred in South Africa. Overall, it seems like the vaccine efficacy drops by about 25 to 30% against the South African variant in particular. So it is good if you're able to start off at 95%, and then if you drop by 25 or 30%, you're still in a good spot. I think one word of caution is that the 95% efficacy that we saw with Moderna, Pfizer, and BioNTech, that's almost like the optimal in the sense that people were vaccinated and then very soon after exposed. And so we were really measuring the efficacy at almost the peak immune response to the vaccine. We don't know what the vaccine efficacy looks like 2 years after vaccination or 1 year after vaccination. We would expect some of the antibody titers, some of the cells to wane over time and maybe be more susceptible to certain variants. But overall, the vaccines do work, whether it's 50, 60% against some of the variants, and it does help stop the spread of the virus, and that should help cut down on the formation of new variants.
So I'll add because was actually included this, and that is, do you acted
And
by
Angela, your audio is
second
all.
dose strategy?
Angela, your audio kind of broke up for the first part.
Oh, okay. Let me turn off my video then. Is that better?
So far, yeah.
We are coming up. Actually, I realized we have a hard stop right at quarter till.
We have a hard stop at in 15 minutes. We have a wind down period. Yeah.
Got it. All right. So let me just ask this follow up then, and that is, do you think that what we just discussed would be impacted by a single dose strategy for the mRNA vaccines or a delayed second dose strategy?
I think a single dose strategy does affect that. I think there's some estimates. Maybe the mRNA vaccines are closer to 75, 80% efficacy after one dose, and the second dose gets them up. So again, if you're starting lower and then you take a drop against the variants, that's suboptimal. Time between doses is interesting. I think from an immunological standpoint, it's probably even better to wait a little bit longer than the 3 to 4 weeks. I think that's almost the minimum time where you'd see a benefit from having a boost. But there's issues if you wait 12 weeks between, then that's a couple of months where you're partially protected. Just also logistically, people forget to come back for the second dose. So yeah, I'm more concerned with the one dose. That's why I encourage everybody to get the 2 doses.
Yeah, I would say that Jason has given exactly the answer that I would have given there. It's possible that waiting longer could improve immune responses somewhat, but the logistical issues and the fact that you are going longer without getting that full protection can really be a problem. And you want to make sure you get that second dose so that you are getting your full protection.
I totally agree with both of you. On to you, Jason.
Okay. All right. So we talked about the variants. I think, let's see, I guess my most recent question, I think this is the last one. When do you think the vaccines will get full FDA approval? And I don't know the answer to that.
So I can talk a little bit about that. For full FDA approval, the FDA had asked the companies for 2 years of follow-up data. Sometimes people are worried that we haven't had that length of follow-up data in this case. And so that has been a question that people have asked about the emergency use approval, which is how the vaccines are currently approved. And this ties into a question that I'm seeing pop up a lot, which is part of why I'm mentioning it this way. The one thing that we don't have, because we don't yet have that 2 years of follow-up data, is we don't have as much information as we could have on longevity of the immune response and how long that immune response will last and sort of whether we might need a booster. That's something that we don't have yet, though we do have safety data as by and large, vaccine side effects happen within 6 weeks following vaccination. And so right now we don't have the information on longevity, and that 2-year follow-up and that longevity data would probably be needed to get the full authorization.
So a couple of years.
Yes, exactly.
Okay. Brianna, Angela, any additional questions do you have?
Well, I have a lot, but
Oh, okay.
I'm not sure if when we enter the wind down period. I think though that maybe one good question to ask is something that I get all the time, and that is, would you personally use the adenovirus J&J vaccine or the mRNA vaccine, and why? Is J&J more traditional? And if so, why did they not just go with that type of vaccine if there's more data on side effects, et cetera? And for me personally, I take any of the vaccines. I just found out today, actually, that in Washington State, where I am, I became eligible as of today. So now I get to start playing the search for appointments game. Ideally, I'd like to get J&J just because I'm moving to Canada in 3 weeks, and I don't know when I'll be able to get my second dose of an mRNA vaccine, but that's more a practical consideration than anything. I will say that a month ago, I had a dream that I got the AstraZeneca vaccine, and I woke up feeling super excited, and then I remembered that the AstraZeneca vaccine is not available to me, and in fact, I have not yet gotten a vaccine. So I think that, what I tell people is what I would do myself, and that is that I would get the first vaccine that's offered to me. None of these vaccines are quote unquote traditional in the sense that they're not a live attenuated vaccine. There's not an approved adenovirus vectored vaccine that was on the U.S. market prior to this coming out, this pandemic. But both of these technologies have been in use experimentally for a long time. The reason why these were used rather than a more traditional approach like an inactivated vaccine such as the influenza shot or a live attenuated vaccine such as the oral polio vaccine is that these are actually a lot faster to make. As Jason was saying before, you can basically make your mRNA and immediately start making that and testing it for the spike protein. Whereas if you were to make an inactivated vaccine, you would have to figure out how to grow up a ton of virus, figure out how to safely inactivate that, and then you'd still have to conduct the same clinical trials to make sure that it was safe and effective. A live attenuated vaccine is even more complicated because then you're trying to make a virus that will infect you but not cause COVID-19, and that's exceptionally difficult to do with a novel pathogen that we've never seen before. So that's why these vaccines have won the race, so to speak. But there are more traditional vaccines in the pipeline. In China, there is an inactivated vaccine that is in clinical use right now. The Novovax vaccine is a protein subunit vaccine that's manufactured with a newer technology, but it is essentially very similar to something like Gardasil or the hepatitis B vaccine, which are more traditional vaccine approaches. But I think that really the reason why these were used first is that they were just the first ones to develop because the technology enabled them to be made and tested faster.
Yes, and just to kind of give my personal perspective on that as well, when I became eligible for a vaccine appointment, I did not, spend any time thinking about, well, which vaccine is going to be available at this appointment, and should I try to find an appointment with different types of vaccines. I was very excited to get whatever vaccine they had. I didn't know until I showed up that day that I was going to be getting a dose of the Pfizer vaccine. And now I, say go team Pfizer because it was in my arm. But I would have been just as excited had any of those vaccines been offered to me.
And I just got the Pfizer vaccine last week. They just showed up and they gave it to me. There's these differences in efficacy, 95, 75. That's just against symptomatic infections. They're all 100% efficacy against severe disease and death. And so that's the main point. That's why you should just take whatever available, because so far they're 100% efficacious against death.
Yeah, And that actually ties into a question that just came in that I thought was maybe a sort of important thing to remind people. Someone asked, my 88-year-old mom just called me with this question, can I still get sick even though I am more than 2 weeks after my second vaccination? And so I think that that's really important to point out. Jason just mentioned that these vaccines were shown to have 95% efficacy against symptomatic disease. And so that means that, yes, there is still a small chance that you can get symptomatic disease. However, they had basically 100% protection against death and a very high percentage of protection against severe disease. So could you still get sick? Yes. But you are less likely to get very severely ill. And you are much less likely to get sick.
So you can think of it as protection against asymptomatic infections, where you get infected but don't feel any symptoms, and then there's symptomatic infections, severe disease and hospitalization and death. And so your immunity is strongest at the bottom of that and goes up. And as your immunity wanes, that's when you'll likely start to have people get asymptomatically reinfected and then maybe symptomatically infected. And then hopefully the vaccines, though, because they elicit antibodies and T cells, will continue to protect against the severe disease. But likely we will need booster shots at some frequency, maybe reformulated against different variants. We're just trying to figure that out, but it's well within the capabilities. The companies can quickly retool the vaccines as needed. All right. Well, maybe we should wrap up just so we don't overlap with the other group. So thanks everybody. This is, I think, one of the largest turnouts that we've had. So thanks so much for spending part of your time with us, and I hope it was informative, and feel free to reach out with any additional questions or attend one of the many other sessions.
Yeah, we apologize for all of the questions that we weren't able to answer, and we thank you all for coming today. Hopefully you can go to another town hall or otherwise reach out to get your questions answered, because these are great questions, and thank you so much for your interest.
Yeah, I'd like to also thank everybody and to just echo what you both just said, that there are going to be a lot more of these town halls. I personally am doing another one on April 5th. I think I've seen that both of you are signed up for other town halls in the future. So if you know somebody who would like to come to a town hall like this, or if you feel that your question wasn't answered, or you have a burning question that you didn't think to ask until later, of course, go ahead and ask on Twitter, but you can also attend any one of these fantastic town halls. Like, all of the people who are giving them are rock stars, and everybody has unique expertise and unique experiences and unique ways of communicating. So, you know, definitely, if this was useful, highly recommend going to another one. But thank you so much for having me, and thank you all for coming and bringing such fantastic questions.
Take care.
Take care. Get vaccinated.
Be well, everyone.